Hello biologists, today we're going to talk about lab 2.13, natural selection, which the specific title of the lab is called Fishy Frequencies because we're going to use some goldfish and some pretzel fish to talk about what happens as natural selection occurs in our fish population. Let's remember that natural selection is the engine of evolution. It's what drives evolution, and in order for evolution to happen, we need three main things. We need variation, that's differences among individuals in the population. There's lots of that in humans, you can see it. Selection, in other words, something's acting in the environment on individuals with differences that makes some individuals more fit for their environment. So maybe some can run faster, maybe some can hide better to get away from predators, others just might be able to find food better or be more successful in reproducing or mating. Those are some types of selection, and all of this happens over a long period of time. So there's three ingredients in evolution: variation, selection, and time. So keep that in mind as we talk about the different elements, the different pieces of natural selection. First of all, let's talk about variation, and I think this is a creature you're all familiar with. Here's a picture of gummy bears, and here's a perfect example of variation in a population. There are different colors of gummy bears. Selection would happen if there's a predator. I have a cute little hedgehog toy here that's representing selection by predators. This hedgehog prefers red gummy bears because apparently they're easier to find on the green environment that this hedgehog is in. So this hedgehog is going to select red gummy bears to eat. Over time, the hedgehog is going to keep selecting those red gummy bears. So the population, as years go by, may change if this hedgehog keeps just eating red gummy bears, or as many reds as it can find. So the population in year 2 might include fewer red gummy bears, just because they're eaten more often, and the hedgehog might have to switch because it has a harder time finding red gummy bears. It might have to switch to its second and third favorite, orange and yellow. So here's, it might just be many years, but over time, hedgehogs are going to keep selecting orange, yellow, and red, and that's going to mean that there are more green and white gummy bears left in this population. So this hedgehog is selecting against the red, yellow, and orange gummy bears because it's eating them, and it's selecting for the green and white gummy bears here because those are the ones that survive. So by year 4, you might be down to just mostly green gummy bears, and the hedgehog might be actually eating white gummy bears because it can't find as many red, yellow, and orange ones. So given what we've talked about with this hedgehog and its selection, which population do you think is going to be left at the end of several years of hedgehog feeding on gummy bear populations? Is it going to be population 1 or population 2? Now we can finally talk about fish. Remember, natural selection changes how many of each type of allele are present for a gene in a population. So here in this population, we're talking about the gene for color in our fish. We have 2 alleles here. We have the gold allele and we have the brown allele. So there's one gene with 2 versions of that gene called alleles. Those 2 alleles are gold and brown. Natural selection changes how many of each type of allele are present for a gene in a population. Remember, let's go over some definitions here so you get these all kind of straight in your head. before we go on and start using them in context. A gene codes for a protein or one characteristic of an animal. An allele is a version of a gene, in this case it's brown and gold, and a population is all the same species of animal living in one place at a particular time. I put the camera here on this group of goldfish, this population of goldfish. You can see some of them are cheese goldfish and some of them are pretzel goldfish. That's representing the brown allele and the gold allele in our population. If you've read the lesson, you remember that there are 3 types of natural selection. There's disruptive selection, which selects for things that are kind of very different from the main or average condition in the population at the moment. There's stabilizing selection, so perhaps if a majority of the fish were brown and that was working for the population, they were able to be camouflaged better. There would be more brown fish in this type of natural selection. And directional selection says whatever's over here is not working. The environment has changed, so this color doesn't blend in anymore, or these animals aren't fast enough to get away from predators. So it's changing the structure or the color or the behavior of the organism, so it's very different from what was currently there. So what is after natural selection is very different from what was before. It's a little bit like disruptive selection, but disruptive selection goes in 2 different directions at the same time. It's over here and over here on both tails of kind of the average. It's not just going in one direction like directional selection. So we're going to see what kind of natural selection is perhaps happening in our population here. So we have a mini ocean, which we saw, full of these types of fish. So we've got gold and we've got brown. And each of these different options, the brown can have 2 different combinations of alleles, and the gold can only have 1. The gold allele is recessive, so in order for the gold phenotype, or what you actually see in the fish, to be expressed, it's got to have 2 little f's. Now brown, you've got 2 options. It can either be heterozygous, big F, little f, because it's a dominant allele and it'll show up even if there's only 1 copy, or it could be big F, homozygous dominant. So keep this in mind. We don't know what the genotype of a brown fish is, but we do know the genotype because there's only 1 of a goldfish. We do know what genes, what alleles the goldfish has, but we don't know exactly what the alleles of a brown fish are. But we can do some math and figure that out. We just answered this question. We don't know for brown, but we do know for gold. You cannot tell the genotype from the phenotype in the dominant case. In other words, you can't tell what genes or alleles the fish has if it's a brown phenotype. But you can figure it out using an equation, and you can figure it out in the gold phenotype. You can see gold and you know there's only 1 option for gold, so you do know the genotype here. So now we're going to do some natural selection in our ocean full of fish. So we have our population of goldfish and pretzelfish, and we're going to pretend we're a hungry predator and do some natural selection. What we're going to do in this lab, and we're going to do a little bit of math, but don't worry, I will help you. We're going to see how the allele frequency changes over time. Remember, allele in this case is big F, which is the dominant allele and ends up with a brown phenotype, or little f, which is this gold phenotype, the goldfish crackers you're very familiar with, I'm sure. So the procedure in the lab says that we should select 10 fish from our ocean. And so I've already done that and taken a picture of it, so it's simpler. And you can see that we have 1, 2, 3, 4 brown fish and 6 goldfish. So we're going to record that data. We have 6 gold and 4 brown. Now here's where the natural selection comes in. So we've taken a subsample from the ocean. That's 10 fish. We know what that is. 6 plus 4 is 10. And now we're going to do some natural selection. We're going to pretend we're a hungry shark and we're going to eat only goldfish. Not brown fish, just goldfish. So the fish we've eaten are going to be replaced from our ocean here full of fish. We're going to grab 3 more random fish. We're not going to peek. And replace those fish. So we have another generation of fish. Here's the picture. Brown fish are hard to catch because they have camouflage. You can see in this bad picture that the brown are blending right in, and those goldfish pop right out. So we have 10 fish again. We have 1, 2, 3, 4, 5 goldfish and 5 brown fish after the shark fed on them and the fish died and then they were replaced from the ocean. So fish are born to replace the ones that are eaten. And we get fish generation 2. So this time we don't have as many goldfish. We only have 1, 2, 3, 4, and we have 6 brown fish. So we're going to record that data in the table. We have 4 goldfish and 6 brown fish. So we're a hungry shark again. We're going to eat 3 goldfish. Nom, nom, nom. We're going to replace those goldfish randomly from the ocean because fish are born to replace fish that die. And we're going to have a new generation, generation 3. This time we only have 3 goldfish left. So in our table, generation 3 is going to look like this. So again, after generation 3, we eat 3 tasty goldfish. Nom, 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 nom. We're the hungry shark. We replace the goldfish that we've eaten randomly from the ocean. You just grab in, dive in, and pick 3 fish. Add them to the ones, the generation that you've eaten from. And now you have generation 4. Generation 4 only has 1 goldfish and 9 brown fish. So you can see natural selection might be having an effect here. In generation 4, something different has to happen because since there's only 1 goldfish, the shark can't eat 3 goldfish. This time the hungry shark, which of course is me or you if you're following along with your own goldfish, has to eat 2 brown fish and 1 goldfish because it can't find any more goldfish. So here's generation 5. The shark ate 3 fish. We replaced them from the ocean by just pulling 3 random fish. And now we have generation 5. And you can see we're back up to 2 goldfish this time. And we're going to record that in our table. So here's our data. You can see how the goldfish changed over time and how the brown fish changed over time. Now we're going to calculate the percentage of goldfish. So if we have 6 out of 10, that's 0.6. So when we fill in the table, we're just going to take how many goldfish and divide it by 10 because that was the number of fish in our sample. So you can see I already did the math for you. Here's 6 out of 6 divided by 10 is 0.6. 4 divided by 10 is 0.4, and I filled in the rest of those numbers. And you can pause and do the same. we can use some nifty math called the Hardy-Weinberg equation to find the frequencies of the alleles. Remember, we were interested in not just the color of the fish, but the genes that, the alleles that cause the color of the fish. Don't worry, we're going to do the math together. It's not that hard. Just take a deep breath. Just because you see some algebra on the screen, it doesn't mean you need to panic. The Hardy-Weinberg equation is p squared plus 2pq plus q squared. Now what that means is basically p plus q, your two options for alleles. You can think of brown plus gold has to equal 1. There's only so many gold alleles, so there can only be so many brown alleles because there's only two options and they have to add up to 100%. So in our case, we can think of it as big F plus little f has to equal 1, or the percent or proportion of big F plus the proportion of little f has to equal 1, or if you think of it percentage, it's like if it's 50-50, it has to equal 100%, or it could be 60-40 or 30-70 or 20-80%, and that would add up to 100%. Because there's only two options, they always have to add up to 100%, or if you're using fractions or decimals, they have to add up to 1. So if we want to find the frequency of the recessive gold allele, in other words, little f, we can just take the square root of the percent of gold fish. So if we take the square root for the first one of 0.6, we get 0.77, which means that 77% of all the alleles in the population are recessive. If we go back to the Hardy-Weinberg equation, this gives us the recessive allele. This gives us the individuals that are heterozygous, and this gives us the homozygous dominant individuals. So this is homozygous recessive. So if we take the square root of this, we know the proportion, or like the decimal version of percent, for this allele. So if we take the square root, we know that 77% of all the alleles in the beginning were gold alleles. So I just took the square root of 0.6, I got 0.77. I took the square root of 0.4, I got 0.63. I took the square root of 0.3, I got 0.54. And you can see all of the calculations I did here. And you can just use my numbers. So this here represents F squared, that's percent of goldfish. That's the phenotype. And we could figure out the alleles or the genotype by just taking the square root. Now we're going to ask ourselves another question. What's happening to the frequency of the recessive gold allele over time? So if we look at these numbers, it goes 0.77, 0.63, 0.54, 0.31, 0.44. What's happening over time? What's the trend in these numbers? I'll let you think about that. Now that we know the frequency of little f, all we have to do is take 1 minus little f equals big F, because we've only got two options and they have to add up to 1. So for the first generation, 1 minus 0.77 equals 0.33 for big F. So 0.33 of the population, the proportion, or 33% of the population, is big F. And I did the math for you here in this table again. And so here, oh, this should be 2.3, sorry. So there's 0.77 and 0.23. There's 0.63 and 0.37. And you can see as we go down, what's happening to the trend here? It's 0.23, 0.37, 0.46, 0.69, 0.66. What's happening to the frequency or the percentage of the dominant brown allele over time? What's the trend in these numbers as you go through the generations? If you want to do this on your own, you can follow the directions on the lab sheet and record your own data in the population and get some extra credit that way. You don't have to have fish. You can use game pieces or... Okay, now that we've gathered our data about our population of goldfish that were eaten by hungry sharks, let's talk about some of the questions on the lab. First of all, let's think about how natural selection and why natural selection took place in this lab. So, what was random and what was not random? Well, if you think about it, here's a hint. The sharks were eating only goldfish. So they were selecting against goldfish because they were eating them, meaning the brown fish, the pretzel fish, were more likely to survive. So these little guys were more likely to survive than these little guys. The sharks were eating these first if they could find them, and then if they couldn't find the goldfish, then they would settle for the brown fish because the goldfish were easier to find. So natural selection happened because of something non-random. The sharks just didn't dive into the population and eat whatever. They were picky. They were just able to find the goldfish more often. And that's why natural selection occurred, because something non-random happened. The non-random thing in this case was that only goldfish were selected and eaten, leaving the brown fish. So, like we said before, the goldfish, the shark here, eating just the goldfish because they were easier to see, was a non-random event. And when something non-random happens, you're likely to get natural selection. So because the shark is eating the goldfish, these brown fish are left to reproduce. So we saw the percentage of brown fish increase and more big F alleles appear. So here's question 3. We need to think about the frequency of the alleles. Remember, in the table here, we looked at the frequency of each allele, or the percentage of each allele in the population. For example, at the beginning, it was 77% gold alleles and 23% brown alleles. At the end, it was 44% gold alleles and 66% brown alleles. So now that we looked at those frequencies, what happened to the frequencies of the alleles during the brief period of our lab? In other words, did we see more big F or little f as time passed? Remember, little f is the allele for the gold color, and big F, the dominant allele, is the allele for brown. Let's think for a second, because a lot of students always seem to think that the dominant allele is somehow always better. And in some cases it is, but here's a great example of where it isn't. These are katydids. They're insects that you can find here in Minnesota. They they live out in green foliage, maybe in your garden. The dominant form of katydids is this bright pink. Now, the reason we don't see a lot of this dominant form, even though it appears the katydids are pink, even if they only have one copy of this allele, which you'd think there'd be more of them, they're not. Because this is a bird snack pretty darn quick, and they don't live long to reproduce. So this allele doesn't survive long in the population. That's pretty crazy, huh? So just because it's a dominant allele doesn't mean it's always better somehow. It's whatever nature thinks is better. And in this case, nature doesn't think that pink, even though it's dominant, is better. Okay, question 4. Imagine that a new species of shark is accidentally introduced into the ecosystem when an oceanside aquarium is hit by a hurricane. So a pair of sharks gets out of the oceanside aquarium because they're released by a hurricane. The new species is going to eat more brown fish because that's what it ate in its previous habitat, and it's just got new tools for finding fish. And so it can find the brown ones better. How might the changes in feeding habits of the shark population affect the population of Keppridge farmish? That's our fish here. That's the scientific name for our species of fish here. In the ecosystem we have been studying. What might happen to the gene frequencies for brown, that's big F, or gold, which is little f? So think about that. If this new fish moves in that can find brown fish better, what might that do to gene frequencies or the percentage of each allele in the ecosystem? So here's a great question to think about because every scientific model has weaknesses. What are some weaknesses in our fish model? Here are some things you can think about. The number of generations we studied, the birth and death rates, are they always perfectly equal? Do real sharks hunt and just find goldfish, or might they just take whatever they can find? Do real fish, no matter what color they are, can they hide? And where do real sharks and real fish actually live? And how might their environment affect the allele frequencies in different ways that we didn't consider in this model? So think about all the things that this model didn't consider and use that in question 5. So here's a couple other things to think about. What would happen if we continued our model for another 5 or 10 generations? Here's an example of how mutations create variation. And unfavorable mutations are selected against. So gold might be, if it was a mutation, might be selected against. Reproduction and mutation occur. Favorable mutations are likely to survive. So that's something to think about. If we continue this model to generation 10, generation 15, generation 20, what might we happen to see over time if we use the same model of natural selection? And what might happen if the habitat changed? If all of a sudden, instead of a kind of brown habitat, we had an orange habitat? How would that affect the gene frequency or the allele frequencies in this lab? Here's a good MCA practice question. What kind of selection would occur if the habitat changed as below? Would we have directional selection? Would we have stabilizing selection, or would we have disruptive selection? So if the habitat changes and the goldfish are able to blend in more easily and they survive and the brown fish are crossed out, what type of selection might that be? Here's one other thing to think about. You can add this to your lab as an extra credit question. If there's a new fish in town, look at this crazy new fish that was introduced by a cargo ship pumping ballast water from another area, which actually happens a lot. That's how zebra mussels were introduced into Minnesota lakes. What do you think is going to happen now? You've got all these colors. How are the sharks going to react? How the fish that are already there going to react? What might happen to the allele frequencies of the population that we have? And what might happen to the allele frequencies of this population of fish that were introduced? Thanks, this is Miss R. Unfortunately, we don't have office hours anymore, but you can make an appointment to see me. You can call me or you can Skype me. Thanks so much, everybody. Bye-bye.